Welcome to Biomandscape. Today we are going to know what are macrophages. What is a macrophage? Macro means a big and phage means eater. So this macrophage, it is a big eater of immune system. It is derived from a Greek word macro, that means a big, and phage means to eat. So the macrophage, they are the large professional phagocyte. They play important role in both innate as well as the adaptive immunity. They are found in almost every tissue. They are long-lived tissue and they reside in the immune cell that arises from the embryonic precursors like the yolk sac and fetal liver. So let's know about the various organs where they are found, and there are various types of macrophages that are present in the human organ at various locations. Like in liver, the Kupffer cells are present. In lungs, the alveolar macrophages are present. In brain, the macroglia cells are present. This macrophages type, and in bones the osteoclast cells are present. In spleen, the splenic macrophage are present. Then peritoneum, in that peritoneal macrophage are present. And though they look different and go by different names, they all function similarly and recognize, engulf and digest and alert. So let's know from where they origin and their development. In bone marrow, monocyte, from bone marrow to monocyte, and from the blood to the tissue, and then finally the macrophages are formed. Most adult macrophages develop from the circulating monocyte which exist in the bloodstream and different under the influence of the macrophage colony stimulating factor. These macrophage colony stimulating factor, they are the key cytokine, and there is a transcription factor PU1. So it have various functions. So in phagocyte, its function is to engulf the pathogen, dead cells and debris, and it is used in the PRR like TLRs and scavenger receptor. Now I have made a video on the various type of receptor. You can visit and watch those video of immune cell receptor. There we will know about the PRR, what are they, and toll-like receptors and scavenger receptor. Let's come to the topic where the fused phagosomes with lysosome, they will form the phagolysosome. Then in antigen presentations also, it have an important role for the process of pathogen fragmentation, which is present on MHC2 and 2 CD4 plus T cells. That is it act as an APC. So antigen presenting cell, they are the important part of the immune system that are important in activating the adaptive immune responses, and they capture, process and display antigen, that is a foreign substance that is present on the surface of the other immune cell, primarily the T cells, less potent than the dendritic cells. Then cytokine production, in that it secrete the interleukin 1, interleukin 6, then TNF alpha, tubonecrosis factor alpha during inflammation, and recruiting the neutrophils and other immune cell. In tissue repair also, in the production of TGF beta, MMPs and VEGF, in that it is important, and it promotes the antagonistics in EMC remodeling. So there are types of macrophages. We can write it here. It is split into two, that is M1 and M2. In that it function in pro-inflammation, microbicidal and anti-inflammation. In the tissue repair, there are various types of macrophage triggering cytokines like IFN and gamma and LPS, and its function is pro-inflammation, microbicidal and M2, M1 and M2. These are the two types of the macrophages, and their roles and functions are given here. So M1 macrophages are like warrior, killing pathogen, producing reaction oxidative system and nitric oxide. In M2, macrophages are the healer, and they suppress the inflammation and rebuild the tissues. So once more about the macrophages polarization, in that it have M1 and M2. These are the two type of path. The M1, it triggers the cytokine, and its function is to pro-inflammatory microbicidal, and in M2 It also triggers the cytokines like interleukin 4 and interleukin 13, and its function is to anti-inflammatory and tissue repair. So M1 macrophages are like warrior, killing the pathogen and producing the ROS and nitric oxide, and M2 macrophages are the healers. They suppress the inflammation and rebuilding the tissues. In that it is important. And then there are various surface marker and receptor macrophages show with multiple receptor labeling like CD14, CD11B, 11 in MHC2. In that it plays important functions like CD14, it function in LPS recognition, in CD11B addition and phagocytosis, in MHC2 antigen presentation, in CD80 and 86 co-stimulation. FC receptor example CD16 binds the IgG in opsonizing the pathogen. For opsonize pathogen, its function is to opsonize the pathogen. Then in various diseases it is important like infection, cancer. In infection, to kill the bacteria like bacterial tuberculosis. In cancer, tumor associated macrophages like TAMS may support the tumor growth. M2 like. Then in atherosclerosis, in that it form the cell, they are a lipid ladder. Macrophage in the plaques. In autoimmunity, overactivation of it can cause the tissue damage like rudimentary arthritis. Then clinical relevance that have like checkpoint inhibitors, reprogramming macrophages in cancer. In nanoparticles, it can deliver drug directly to the macrophages. So macrophages, they are large to eat, and macrophages means large to eat a specific phagocytic immune cell that plays a role in the innate immunity, inflammation, antigen presentation, and tissue homeostasis and repair. So in its origin and development in various stages, we can see like embryonic stage, postnatal stage, and monocytes. In embryonic stage, it is derived from the yolk sac and fetal liver, which form the tissue residing the macrophages. Then in postnatal, it is derived from the monocytes of the bone marrow. In monocytes, first it is circulated in the blood, then enter the tissue, and then differentiated into macrophages, monocytes. Then the growth factor which are required for this macrophages are MCSF, that is the macrophage colony stimulating factor, and GM-CSF, that is in the inflammation, and interleukin 34 in some tissues, and the key transcription factor which is used is PU1. And as we all know, it is tissue specific type, like in brain it is microgliae, in liver it is Kupffer cells, in lungs it is alveolar macrophages, in bone it is osteoclastis, and in spleen it is red pulp macrophage, in peritoneum it is peritoneal macrophage, and in skin it is Langerhans cells, and also considered dendritic. So these are self-renewable and populations and seeds during the embryogenesis. So there are various functions in phagocytosis, like toll-like receptor, it detect the PAM, in scavenger receptor, in mannosol receptor, in Fc gamma receptor like CD16 for ADCC, and complement receptor like CR1 and CR3 in that. Then it fuses with the phagosome plus lysosome, which form the phagolysosome. That is, the fusion of phagosomes plus lysosome will form the phagolysosome, and after that it will get digested using the ROS, NO and enzyme. Then antigen present in that, MHC class 2 molecule present the antigen to CD4 plus anti-helper cell. There is a co-stimulatory molecule like CD80, that is B71, and CD86, that is B72. It is less efficient than dendritic cells but still efficient. Then there is cytokine production, key pro-inflammatory cytokine like interleukin 1, interleukin 6, TNF, alpha, interleukin 12, and interleukin L10. The interleukin 1, it is important in the fever induction, leukocyte recruitment. interleukin 6 in acute phase response. Then NF alpha inflammation activities and endothelial cells. Interleukin 12 is used in the activates the NK cells and Th1 differentiation. Interleukin 10 is used in the anti-inflammatory cell. Then tissue repair and remodeling, in that TGF beta, that is transforming growth factor beta, VEGF, that is vascular endothelial growth factor, MMPS, that is the matrix metalloproteinase that promote the antagonistics collagen remodeling. Then there are various types of macrophage polarization, like macrophage 1. It triggers the IF alpha, IF gamma, LPS and TNF alpha. Its function is pro-inflammation microbicidials in production of interleukin 1, interleukin 6 and TNF alpha and ROS and NO, in that its role is to kill the bacteria and activate Th1. The marker which are used are CD80 and CD86 and INOS. Then M2 macrophage, interleukin 4 and interleukin 13, in that its function is to anti-inflammatory and tissue repair in production of interleukin 10 and TGF beta and in the growth factor. The role is to promote the healing and suppressing the immune response, and the marker which is uses are ARG1 and CD206. So its role in the disease is to bacterial engulf and destroy the pathogen, to limit the speed of viral infection and to remove the debris and promote the regeneration like wound healing. In that it is important. Then pathological roles it have like chronic inflammation, for that in excess of cytokine production and atherosclerosis form the macrophages plus oxidize LDL. In cancer, tumor associated macrophages may promote the tumor growth like M2 like, and tuberculosis, in that microbacteria survive inside the macrophage. And in autoimmunity, macrophage overactivation damages the tissue. Example RA. So the next one important in macrophage is the clinical relevance. So the monoclonal antibody, in that a macrophage, the body's ultimate clean up the crew, and are not just defenders, they are also becoming a powerful tool in modern medicines. So for cancer immunotherapy to infectious disease control, let us explore how the clinical science is harnessing the immune giants. So the monoclonal antibody and antibody binding to the tumor cells, macrophages approaches and release the toxic granule. First, the monoclonal antibody. These label-made antibodies are designed to target a specific antigen on cancer or infected cell. Once the antibody binds to its target, the Fc region remain exposed, and this recognized by the Fc gamma receptor like CD16 on a macrophage. So this interaction trigger the antibody-dependent cell-mediated cytokine, where the macrophage engulf or kill the target cell. So the clinical example is rituximab tagging CD20 on a B cells in lymphoma. Then there is a checkpoint inhibition, reprogramming macrophage in cancer. So in the tumor microenvironment, macrophage often get hijacked, and these become tumor associated macrophage TAMs, mostly of the M2 phenotype, which promote the tumor growth, antagonistics and suppress the T cell. Then there is a checkpoint inhibitor originally developed from the T cell and now be exposed to reprogram the TAM back to M1 like pro-inflammatory cell, and this shift macrophages for being tumor protector to tumor destroyer. Example, it targets the CSF1R, a survival signal for TAM is under clinical trait. Then nanoparticle in drug delivery to macrophages. So in nanotechnology is revolutionizing the drug delivery, and macrophages are the key target. Why? Because they naturally phagocytes foreign particle, include engineering nanoparticle. By coating the nanoparticle with ligands, it recognized by the macrophage receptor like the mannosol and scavenger ligands, drug can be delivered directly inside the macrophage and released slowly over time, and even programmed to switch the macrophage behavior. So using in inflammatory disease, cancer and even brain deliver using the microgalli. Then it is also used in the vaccine. So vaccine particularly being taken up by the macrophage, and these are being used to boost the vaccine efficiency, and especially for intercellular pathogen like the TB, that is Mycobacterium tuberculosis and Leishmania. By designing macrophage targeted vaccine carriers such as the liposomes, microparticle or adjuvant that activate the PRR, pattern recognition receptor, we can enhance the antigen uptake, cytokine production and T cell activation. New TB vaccine platforms are used, and this approach to replace or boost the BCG. So let's have a quick recap about the monoclonal antibody. Their role in the macrophages is engaging the Fc gamma receptor for ADCC. Then checkpoint inhibitors reprogram the TAM2M1 phenotype. Nanoparticle deliver the drug by phagocytes, and vaccine enhance the antigen uptake and presentations. So macrophage are no longer just generators of the immune system, they are becoming precise tool in immunotherapy, drug delivery and vaccination. So this versatility make them a prime target for the future medical breakthroughs, and stay tuned for next video. Thanks for watching. Do like, share and subscribe.